Hello everyone, welcome to this session. My name is Nikhilesh Yadav. I am a PhD research fellow in the area of information systems at Indian Institute of Management, IIM Indore, India. I am also currently working at CSIR National Chemical Laboratory, Pune, India. Today I am going to present my work on topic Pin2DB, a manually curated database of Pin2 type protease inhibitors, which is a part of my ongoing research at IIM Indore. Development of a wide range of high-throughput experimental methods and computational efforts have led to an explosive growth in the biological data. If not properly managed, it will mean waste of resources and the efforts put in generating these data. Hence, it is important to develop biological databases which would help in collection, organization, analysis, and sharing of biological data. Now, following are some of the widely used biological databases which captures the information about various protein sequences. Now, one of the protein types are the plant protease inhibitors. Now, plant protease inhibitors are small proteins which are found in plants. Now, these help in protecting plant from insects and the pests. Now, one of the important family of these plant protease inhibitors is the Pin2 type plant protease inhibitors, which are also known as potato type 2 plant protease inhibitors. Now, in spite of their important role in the plant defense mechanism, the Pin2 type protein family remains unexplored due to lack of annotated protein sequences in currently available databases. This was one of the motivation behind developing this biological database for this family. Now, the other motivation was the presence of the expertise in the form of scientists at the Biological Chemical Sciences Division at CSIR National Chemical Laboratory, which are working on this particular Pin2 type family for the past many decades. Hence, the primary objective of this research work is to create a biological database of Pin2PI family with currently available resources. Now, this database is known as Pin2DB and it would serve as a major resource for Pin2 plant PI's research on which data exploratory data analysis can be done using statistical and visualization tools. Now, the methodology which was followed for this work comprises of data compilation, pre-processing and integration, database creation, and information extraction. Now, data compilation involved extraction of data from external sources, the in-house data generation, and extraction of information which is published in various publications. Now, second, pre-processing and integration ensures the data correctness and completeness. And finally, the database is created. And once the database is created, the information extraction can be followed by using data mining and visualization techniques and by developing a web-based information system. Now, the development model which was followed for the building up of this biological database of Pin2 type PI family is depicted in this slide. On the extreme left shows the four blocks which shows the data sources from which the data is extracted for this biological database. Now, the basic information about the 414 Pin2 PI sequences were extracted from the UniProt database. Now, IRDs, which are also known as domains, are 50 to 55 amino acid long proteins. Now, the IRDs for the Pin2PI were extracted from HMMER website and about 691 IRDs were captured. Now, each IRD, that is the domains, contains a tripeptide loop, which are also called reactive center loops or the RCL, which provides the target specificity or in the layman language the functionality to the Pin2 type PIs. Now, the data of these 63 RCLs was prepared in-house at the Biological Chemical Sciences Division of CSIR National Chemical Laboratory. Now, a single Pin2 type PI consists of multiple IRDs or the domains which are joined by 5 to 6 amino acid regions. Now, these 5 to 6 amino acid regions are known as the linker regions, and the data of about 78 linkers was prepared in-house at the Biochemical Sciences Chemical Divisions at CSIR National Chemical Laboratory. Now, To capture the information from the data extracted from the different sources, it needed to be pre-processed and integrated. The pre-processing of the data was either done manually, or in many cases it was automated by using Python programs or via structured query language queries and procedures. Now the middle section of this slide shows the pre-processing section and gives details about few pre-processing which was done for this particular database. For example, for locating the RCL in domains, finding domains in the sequences, finding linker regions in sequences, and calculating various properties like amino composition of the sequences. For each of these pre-processing tasks, either the Python programs were written or structured query languages procedures were used. Finally, the result of this pre-processing and integration and step was the creation of the Pin2PI database, on which in future data analysis can be done using data visualization techniques and data mining techniques, and also a web-based information system was developed on this Pin2PI database. Now, the result of this work was the creation of the Pin2DB database, and this slide depicts the database schema of the Pin2DB at the conceptual level. Now here we shows the various entities involved in the schema by the various colored rectangles. Now the red color rectangle represents the primary entities of the Pin2DB database, which are the Pin2PIs and the domain. The green color rectangle represents the entities which provides the detailed about the primary funds, and the gray color rectangle represents the entities which provides the support data. Now, this particular slide gives a bird eye view of the overall Pin2DB database. Now, till this date, the Pin2DB database captures functional annotations of about 415 Pin2PI sequences, which are distributed across 109 species. Now, also it captures information about 698 domains or IRDs and 63 reactive loops, that is the RCL, and 78 linkers. Now this table also shows the distribution of the Pin2PI sequences and the reactive loop RCLs as per their target specificity, and also the distribution of the domains and the linkers according to the various types are also depicted in this table. Now in the extreme right column of this table, the distribution of the Pin2PIs across the 14 domain architectures found in the Pin2DB database is given. Now, domain architectures are the various patterns in which the domains are found in the Pin2PI sequences. Now, beside the Pin2PI database, which was the output of the study, the other major output of this study is the website which was developed for the public access of the Pin2DB database. Now, this website can be accessed using the URL https://pinir.ncl.res.in. This website provides an intuitive and user-friendly web interface to browse, analyze, search, and download information related to Pin2type PIs. Now, this website comprises of four major modules, which are the browse module, which provides the facility of browsing the Pin2DB database. Then second is the analysis module, which provide a comprehensive analysis of the Pin2DB database. Then there is a search module, which provides the facility for searching the information captured in the Pin2DB database. And finally, there is a download module, which provides the users with this facility of downloading the data which is captured within the Pin2DB database. So overall, the major output of this work was the Pin2DB database and the web information system, which was developed for providing the public access to the Pin2DB database. Now, in future, we plan to develop various visualization tools for the exploratory data analysis of the Pin2DB. Now, the major avenues where the visualization tools can be developed are for browsing the various features of the Pin2PI sequence. A feature browser scale can be developed. Apart from that, to a hierarchical display can be developed for depicting the biodiversity of Pin2PI sequences. Also, visualization tools are planned to be developed for displaying the amino acid composition of the Pin2PI sequences which are captured in the Pin2DB database. Apart from this, also data mining techniques would be applied to the Pin2DB database to bring out novel categorizations of Pin2 type PI data, and further prediction model would be developed to predict the various categorization which would come out by this exercise. With this, it brings to the end of my presentation. I thank you all for giving me an opportunity to present my work. Thank you.